Hello everyone. Since practical of biology of class 12 is going on, so myself Mithlesh Kumar Sinha. So today I'm uploading some important VIVA question which may be asked by external. And the question is, first question is, what is gene? And you know that gene is the basic functional unit of DNA. Gene decides the traits of any organism like eye color and tallness, dwarfness. Second question is which enzyme break the fragment of DNA? So the answer is restriction enzyme that breaks the fragment of DNA. Third question is how many types of restriction enzyme? So there are two type of restriction enzyme, exonuclease and endonuclease. Exonuclease cut the DNA at their end like 3 prime or 5 prime side, whereas endonuclease cut the DNA in the middle part of the phosphate. It will break the phosphodiester bond. Next question is name an organism which show the cell division as well as reproduction both. Answer is amoeba which show the binary fission when one cell will divide in two cells. Next question is what is cytokinesis? So answer is division of cytoplasm is called cytokinesis because you know the cytoplasm is jelly-like structure surrounding the nucleus inside the cell which will divide. The next question is what is karyokinesis? And division of nucleus is called karyokinesis. Next question is name the four whorl of flower. So the answer is the four whorl of flower is calyx, corolla, androecium and gynoecium. And you know the collection of sepal is called calyx and the collection of petal is called corolla. Next probable question is name the two characteristics of pollination by wind. So the pollen grain should non-sticky, it should dry and long stamen and stigma should be there. Next question is name the characteristics for pollination by insect. So the flower should be colorful and it must has fragrance also. Next question is which is very important. Name the embryonic stage of development of fetus in human female. So you know that zygote form and after the cell division of zygote blastomere is formed and after the cell division of blastomere and there is a trophoblast and inner cell mass and that will broken down and it will form the blastocyst. Next question which is also very important, what is pedigree analysis? Pedigree analysis is the inherited characteristics that transfer from parents to offspring in terms of disease also that needs to be studied and checked it. Next question is how does ringworm is formed? Ringworm is formed by fungi and type of fungi is microsporum, trichophyton and epidermophyton. Next question is why detergent is used in isolation of DNA? This is also very important question. And you know guys that detergent contains the enzyme like protease and lipase which helps in digestion of protein and lipid and it breaks down the cell membrane also and that is why the detergent is used in isolation of DNA in the practical. Next question is why chilled ethanol is used in isolation of DNA. Chilled ethanol or ethyl alcohol is used to get the precipitate of DNA. Next question is what is the role of sodium chloride in DNA isolation? And you know that sodium chloride is a salt which neutralizes the negatively charged DNA. And you know that DNA is a negatively charged ion. Next question is what is the charge of DNA? That we have already discussed. The DNA is a negatively charged particle. Uh, Now next question is which part of cell contains DNA? And you know that DNA is contained in the nucleus of the cell. Next, which is very important question, what is population density? So population density is quadrant by quadrant method, total number of individual in all quadrant divided by total number of quadrant. Next question is how population frequency is calculated. So population frequency is calculated is that is equal to total number of quadrant in a species divided by total number of quadrant in a species. Next question is what is mitosis? So this is very easy question. Equational cell division of chromosome is called mitosis. Another very easy question, what is meiosis? So reductional cell division of chromosome is called meiosis, which generally occur in the reproduction. Next question is, write four phase of mitosis. So the four phase of mitosis is prophase, metaphase, anaphase, and telophase. Now next question is, which stage of mitosis is best for chromosome? So metaphase is the best stage of mitosis where all chromosomes are in the same line. Next question is, name the stain used for identify the plant cell. So answer is safranin is used to identify the slide of plant cell. Now next question is, what is the feature of prophase? So the feature of prophase is condensation of chromosome fiber into chromosome and nuclear membrane and nucleoplasm that starts to disappear also. So the next question is, what is characteristics of metaphase? So the characteristics of metaphase that chromosome will move to equilateral line. Next question is two feature of anaphase. So the two feature of anaphase is centromere splitting and sister chromatids separate and move to their each equilateral line. So next question is, what are the characteristics of telophase? So sister chromatid reach to opposite pole and reappearing of the nucleus as well as nucleoplasm also. Next question, which is very important question, what is pollen germination? Pollen grain absorb the nutrient from surface of stigma and it will germinate further, and this process called pollen germination, and in practical we will use the sucrose for pollen germination also. Next question is, which is very important, that what is the two layer of the pollen grain? So outer layer is called exine and inner layer is called intine. So how many stage of pollen grain? There are two cell stage. One is vegetative cell, another is generative cell. What is three cell stage of pollen grain? So answer is one vegetative cell plus two male gamete. To put together the three cell stages there. Next question is, what are the characteristics of air pollination? So air pollination, the process should be, the plant should be colorful, colorless, odorless, and nectarless. These are the characteristics of air pollination, which is also called anemophily. Next question is, what are the characteristics of water pollination? Water pollination, which is also called hydrophily, where the plant should be colorless, odorless, and usually nectarless also. And there are two type of hydrophily. One is epihydrophily, another is hypohydrophily. An example of epihydrophily is hydrilla, and Zostera is the example of hypohydrophily. So next very important question is, uh, on which species of pea plant Mendel has done the research, and the right answer is Pisum sativum. Next question, which is very important, that the outer layer of pollen grain out is made of, so it is made of sporopollenin, and inner layer is made of cellulose and pectin. Next question is, what is symbiosis? So symbiosis is the combination of algae, fungi, and lichen where all three get benefited to each other, and this relationship is called symbiosis. Next question is, what is mycorrhiza? Mycorrhiza is a relation between the roots of higher plant and fungi, and this is called mycorrhiza. Next question is, what is homologous series? What is homologous? So homologous are those organisms whose origin is same and but the function are different is called homologous. Next question may be asked, what is analogous? And analogous is just opposite to homologous where origin is different but function is same. Next question is, what is BOD? BOD, it is called biological oxygen demand. The oxygen required by bacteria to break down the organic matter is called biological oxygen demand. If there is more biological oxygen demand, it means that water is more pollinated. Next question is what is PBR 322? PBR 322 is the plasmid of the cloning vector of E. coli. Next question is, and it has two uh, marker gene, one is ampicillin, another is tetracycline. Next question may be asked, what is vasectomy and what is tubectomy for reproduction? So the process of cut and tide of vast difference is called vasectomy, and the cut and tide process of fallopian tube is called tubectomy. So these were some of the important questions which I have discussed with all of you. So thank you very much to all of you.